Hello and welcome to the press conference for the joint meeting between the Acoustical Society of America and the Canadian Acoustics Association. I'm Maura and I will be hosting this press conference today. We'll have presentations now at 11 and later at 2 p.m. During this session, we will have 3 presentations, and after each, there will be time for questions. If you're joining us online, you can put your questions in the chat. Our first presenter of the day is Catherine Eltz from the University of North Carolina at Chapel Hill. She will be presenting about gas emboli in turtles. Okay, Catherine.
Hello everyone, my name is Catherine Eltz. I'm a first-year PhD student at the University of North Carolina, and today I'm going to be presenting my project on the analysis of gas evolution in the hearts, livers, and kidneys of bicaught sea turtles. And this project is in collaboration with the Oceanographic Research Foundation in Valencia, Spain, which is a sea turtle hospital and a conservation research center. So gas emboli form in sea turtles after they're bicaught and their normal diving activities are interrupted. So the sea turtles can swim into or get caught in a fishing net that's pulled behind a boat. And then the sea turtles are pulled up to the surface at a rate that is different than what they would normally do. And the result of this is the gas that is dissolved in their tissues exits in the form of bubbles that enter their bloodstream. And this can cause paralysis and death in sea turtles. If a sea turtle capture is reported, it is brought back to a place like Oceanographic, and the sea turtles are treated. One way that they're treated is in hyperbaric chambers, just like human divers who get the bends. And in the past, the gas presence in bicaught sea turtles has been observed with X-ray and MRI, as well as ultrasound, but to a lesser degree. The image on the screen is a sea turtle at the Oceanographic Center getting an ultrasound, and as you can see, they have to avoid the shell and go into the fin of the turtle. And the gap that I hope to fill with this project is that there's never been a study that shows how the gas evolves over time in the sea turtles, so we are using ultrasound scans of the hearts, livers, and kidneys of sea turtles who had been bicaught to analyze this. So with this project, I hope to combine many different types of information that I gathered by working with the veterinarians in Spain. The first type of information is the time delay between when the turtle surfaced and when the first ultrasound scan was taken. So the turtles are either scanned on fishing boats or at the veterinary center, so there is a difference in time delay there that I wanted to investigate further. I also wanted to look at the amount of gas present, and I did this in 2 different ways. The first is with grades that were assigned to each scan by the veterinarians, and the grades go from 0 to 5, where 0 is no gas, and 5 is so much gas that ultrasound cannot penetrate into the turtle and a clear image cannot be taken. And finally, I wanted to look a little deeper into the actual ultrasound scans themselves and find quantitative metrics that I could extract from the data. And by combining all of this information, the goal was that we could examine how gas evolves over time per organ for the very first time in sea turtles. And this will help us towards our larger goal, which would be to develop a methodology to predict outcomes and determine the best treatment based on the state of gas at one point in time, instead of needing to do a longer analysis. So now I'm going to show some ultrasound scans of these different organs. The first is of a grade 4 in the heart, which is the second highest grade possible. And in the green circle, you'll see a one chamber of a turtle heart pumping some very bubbly blood, and bubbles look like little white dots, and so that's what we're looking at here. The next is a liver scan. Livers are much more stationary organs, so what we're looking for here is small bright white dots embedded in the tissues of the liver. And finally, I have a scan of an organ that's near a kidney, also a grade 4, and what we're looking for here is flowing bubbles through the blood with a blue arrow, and what we call a comet artifact with the yellow arrow, which is when gas causes a bright white stripe to go down the cone of the ultrasound scan. And these are all examples of things that we either have already quantified or hope to quantify in the future. So getting into the results, the first thing that I wanted to do was look at those two groups I mentioned earlier. So turtles are either scanned on the boat or the turtles are brought back to the veterinary center and scanned on the shore. So I calculated the time between surfacing and first scan of all 100 turtles that I looked at in this project, and I was able to find a significant difference between the two groups. So there was a longer delay between the surfacing time of the shore group versus the boat group, which is to be expected, but this was the first time that we were able to prove that there was a difference, a significant difference. And this is important because then going forward, I could look at the different organs according to what group the turtle that was in. So I separated all of the first recorded heart, kidney, and liver grades, and I plotted them. And I found not only a significant difference between the shore and the boat group in every instance, but something very interesting is that the first recorded kidney grade, the median, was a grade 1 in the shore group, but in the shore group for the liver and the hearts, this was a grade 0. And this leads us to the assumption that gas evolves differently in the kidneys when compared to the hearts and the livers. And finally, some results from the brightness quantification of these images. I looked at regions of interest in the heart and the liver, and we see in both cases a general trend that is an increase, and then the brightness decreases at the end. And we attribute this to what we call shadowing, where ultrasound cannot travel through the high amounts of gas in the animal and results in a actually much darker region underneath high amounts of gas. So that's why in the higher grades where you would expect more gas, it actually is darker. And in the kidney scans, we usually looked at a vessel instead of a broader area like in the hearts and the livers. So the only things that we're examining in the kidneys are blood, or if it's present, gas. And so that's why as we see an increase in grade, we see an increase in brightness range. The limitations of this are that sometimes the settings of the ultrasound scans were different between animals or between organs, and this is something we hope to address in the future. So in conclusion, this project was the first time that we were able to show a change in gas amounts per organ over time, which is very exciting. And all the limitations of this project were related to the fact that this data was collected for a veterinary purpose, so on a fishing boat or of a live moving turtle, and so it caused some limitations. But we hope to address these in the future by working on either standardizing the collection methods of this data or in post-processing with image processing methods. So I'd like to thank my home lab at UNC and my PI, Dr. Papadopoulou, and also the collaborators at the Oceanographic Research Department for including me in this project and giving me this great opportunity. Thank you.
Okay, thank you so much. So now we'll take some time for questions. I will actually ask the first question. So my first question for you is, how are the turtles treated once they're found to have this gas emboli?
So the turtles are taken back to the center and given like a kind of general veterinary checkup, including vitals and if they have any like laceration. Sometimes there's other problems that arise from being caught in a net. And then if they have gas emboli present, they'll be recompressed in the hyperbaric chamber that I showed and then monitored for time after that as well.
Thank you. So now we'll look to the online audience. Are there any questions prepared?
Yes, we have a few. The first question is for Catherine. How did you become interested in sea turtles in general?
Thank you, that's a great question. So in undergrad, I minored in marine science and I majored in biomedical engineering. And I saw that my PI, Dr. Papadopoulou, was doing research related to scuba diving and decompression sickness in humans, and she got to know me and my interests and put me in contact with the scientists at Oceanographic, and then they included me in their project. So,
thank you for sharing that. Let's see. From Richard Lovett, we have the question, would similar methods be of any use in humans?
Yes, definitely, and that's one of the big parts of this project is something we call comparative physiology. So learning more about how other diving animals avoid getting decompression sickness can help us in human diving research, and also some of the image quantification methods and the graphical user interfaces we created to help sort through this data can help with human diver data processing and organizing as well.
Well, that's exciting. The next question I have is from Jessica Sansarin. She's wondering, I think you mentioned that your research was done in Spain, but were there other places in the world that turtles are caught this way and research is being done?
Yes, so the turtles that I examined in this project are caught either in Spain, Italy, or Brazil, but turtle bycatch is a problem anywhere where there are turtles and there is fishing. But the data from this project was collected in many different ways. But all of the data in the shore group was taken at the Spain Oceanographic Center. So.
All right, just two more,
Okay.
I think. How could this information be used to better treat turtles in the future?
So the larger goal of this study is to understand both what the high presence of gas looks like and also how it evolves over time. So the end goals of this project are to be able to give a turtle a scan and knowing how long it has been since the turtle surfaced and what the gas looks like per organ at that time, we would be able to predict, oh, it seems like this gas is based on previous trends, still going to rise, and the turtle should be treated with hyperbaric oxygen, or it seems like the turtle has been able to off-gas some of the bubbles on its own, and it would be okay to release the turtle back into the ocean as it is.
I think this question may speak to what you just were saying, but it's from Richard Lovett as well. But does this data alter treatment, or is treatment based on visible symptoms?
So this data currently doesn't alter treatment, and I think your second question was, does current treatment depend on the
Visible symptoms.
visible symptoms? As far as I know, the turtles that are taken to the center are given some sort of imaging to determine how severe the gas is, and then given treatment based on that. So right now it doesn't alter treatment, but that's the end goal of this project.
All right, and the last one from our online audience, are there other animals that this type of research could apply to?
Yes, definitely. In the past, research has found gas emboli in diving mammals, including whales and dolphins, and these gas emboli were found in already deceased stranded whales, and it was interesting because those whales were present in areas where there was a lot of sonar activity underwater, and so that kind of leads us to the assumption that an interruption in natural diving mechanisms in diving animals can cause gas emboli and symptoms similar to decompression sickness. So it's definitely an area of research that can apply to other diving animals. Thank you so much.
Do we have any questions from the audience before? Okay. Thank you so much, Catherine. That was a really fascinating presentation.